me thank the organizers for the invitation and for the chance to leave my country and travel after 2 years that I've been stuck in a rather small place. Right, so let's get started, I guess. So, yeah, so I changed the title of the talk by just a little bit. I added the word experiments because I think that one of the interesting things that has changed recently is that we went from very abstract, comparatively abstract theories to actually seeing these things in experimental data. And so I want to try to make that connection today. So yeah, so we'll be talking about diverse ecological communities. And by diversity, as you all know, well, the first thing everyone thinks about is you have an ecological community, an ecosystem, and it has many species. So you go to a rainforest in India, or you go to Wikipedia, and it says that it has 722 species of flowering plants in 122 families. And actually, that's fancy, rainforest. But if you go here, this is from the south of France, a picture I took. There's 10 species or something like that, of grass just lying around out here. So, it's everywhere. But it's not just the number of species that we think about when we think of diversity. It's also, the number of different chemicals that, bacteria exchange, survival strategies, species associations, and so on and so forth. It seems like everywhere we look and from every direction we see this great richness and diversity. And on the other hand, a lot of the classical theories in ecology describing or trying to describe these rather complex things that we see do try to simplify things a lot. And so let me give you a few examples. One of them is known as neutral theory. Well, okay, we said we had 720 species of flowers, but maybe functionally it's like having just one flower of a few different colors. Maybe it's just one flower for every functional purpose. And then, then if you're colorblind, it's really a much simpler problem of dealing with just one species. And a related question is, if I have a network and I have here, it's actually a measured network of interactions of flowers and insects and a bunch of other things. Well, maybe all I need to do is take all these flowers and think of them as one flower, and all the insects and think of them as one, and all the parasites and think of them as just one parasite, and that really simplifies things. Another family of simplifications is what I call here single axis. That's just a name I made up, or a few axis. For example, a niche trait or some other trade-off, birds that like to live at different heights in the tree, and many examples like that. Species that have more offsprings or invest more in their offspring, things like that. Going to dynamics now, here's an example of you see the abundance, the amount of phytoplankton in some lake as a function over the years, and you see that it's fluctuating by quite a bit. And then, maybe it's due to something, fancy, chaotic dynamics that I will be telling you about in a second, or maybe it's just that the temperature or whatever, nitrogen going into the lake, stuff like that, changed over the years, and it's just a simple issue of external conditions. Or maybe it's 3-4 species that are doing some dynamics, and the rest are just enslaved to this. So even if I have, I don't know, suppose I count 100 species here, maybe it's not rich in the sense that these 100 species are doing something interesting. Right, so the big question that we've been asking is, how do we identify and understand ecological communities that are not one of the above? Okay, so I'll be calling them, I don't know, Haidimensionality, or choose your favorite name for that. And the interesting thing is that it's this idea of having these many variable phenomena, these emergent many variable phenomena, which are not these, few-dimensional mechanics or few-dimensional dynamics, this is clearly seen in other fields. Okay, in physics, you have water melting. That has to do with many dimensions, many variables. In neural networks, flocking, all these things are clear examples of this. And in ecology, it's harder. Okay, for some reason, it's harder. One of the reasons we're already seeing here this issue of external conditions that are really messing up with our system. Okay, separating what is external and what is internal is one of the issues that in ecology is tougher than in other fields. All right. So that was one perspective on what I want to talk about. Another perspective is fitting this within what people are actually doing to study, even in practice, ecological communities. For example, people who are interested in, fisheries. They want to know if if they'll have enough salmon and what they have to do to keep the amount of fish in some area. And so they have this complicated network of who's doing what to whom and so on. And one thing they do is they actually do complex simulations and then think about fitting parameters. What data do you have? Serious issues there. Another thing, again, is related to what I said before, is that really traditional ecology has used these few species theories, things like predator-prey cycles, competitive exclusion, for those of you who might have heard about these things. But then it's hard to systematically connect these two points of view or perspectives of this very complicated network and these theories. All right, so we're trying to do something which is neither of these two approaches. Good. Okay, so first of all, let me say at this point that I very much like and encourage questions during the talk. It helps me as well to gauge where we are, where you are. So please, so I'm stopping now. Anything so far? No, okay. Good, so this is, generally I'll be talking about two themes or two subjects, which are, as you'll see, closely related. One of them is really going to follow things those of you who have done some statistical physics will find very familiar, these ideas of having variables like temperature and pressure, which is all you care about in a gas, or phase transitions, things like that. And another issue, which will be more ecology, let's call it, is these two themes that keep popping up. One of them is randomness. We heard about this issue of randomness even in the previous talk, but here it has to be married with coexistence. Okay, so how do I have something which I think of as random, just to throw out a lot of details, but how do I still combine it with the functionality, if you like, which here is coexistence. Okay, in both cases, I will be trying to lead the discussion towards experiments. So I'll show you two specific comparisons with experimental data. And that's how I'll choose of the many subjects. That's one of my guiding principles. All right, so one slide about this subfield of ecology known as community ecology. This is what we'll be talking about. So of course, ecology is many, many things, right? Nitrogen cycles in the planet, global warming, the economy of natural parks, whatever. In community ecology, what we talk about is, groups of species, of different species, they coexist. And of course, this is very, very broad and varied. But there are some processes that are in common in these many different communities. One of them is motion of organisms, which might be an organism, the bear walking, or it could be that a plant is spreading seeds that disperse through rivers and wind. Another thing that happens is that, that organisms interact. They don't live each separately there. Interactions are very important. Here are some classical examples of interactions. On the right side, you can see interactions. Classical things are competition over resources, different plants, competing over water and sun and so on, and nutrients, predator-prey interactions, mutualism, the bee and the flower helping each other. And there are many, many, many other exotic things that you actually see out there. I like these ants that cultivate gardens, for example, as just one weird thing that one sees. The third thing is that there's always evolution in the background. Evolution in the sense of adaptation, speciation, and so on. I won't talk about that. There's enough for us to handle even without that in the background, although, of course, it is there, and definitely it's not necessarily that there's a separation of timescales. Maybe in certain organisms there is, in microbial populations, these things might be mixed. I'm separating them just for the sake of our discussion. Okay, so what is the simplest setting that would include these two things, motion of organisms and interactions between organisms? Sorry, just a second. Where do I see the time and exactly until when? This is just...
I'm in charge of the time, I guess, so you have until 12:30.
Okay, great, and I see the time here.
11:30.
Okay, it's 11:13. Okay, good. Okay, so what's a very simple model that includes these two things, motion of organisms and interactions? Okay, then the simple thing is, let's take a region, and there's what's known as a regional species pool, which is just the species that are in that region and are there. And then there's a local area somewhere, and these species from this pool move in. Trees, again, distribute their seeds, animals just walk inside. And then there's a local community that forms that has some subset of the species. This process is known as assembly. Okay, that's a community assembly. It's a very important process in community ecology. Okay, and this is the simplest setting, and I'll again focus on this setting. There are others, for example, full account of space, which we are working on a lot, but I won't talk about too much. All right, so now I want to really give you the simplest textbook, classical, canonical example of an equation of dynamics that describe this setting here. And so I'll take one example of the possible dynamics you could write down. It's known as the Lotka-Volterra model. Many of you, I imagine, have seen or heard about it before. So what do you want? So let's start with one species. So if I have one species, so for example, here on the right, you can see the population of sheep in Tasmania. So they were introduced in the early 1800s, and you see that this is the y-axis is the number of sheep in thousands, okay? And you can see, and the x-axis is time, and you can see that initially the population grew exponentially, and then there was something happening there, and at longer times it just saturated to some value, okay? That value is known as the carrying capacity, and so the simplest description that you could write that would do both the exponential growth and the saturation is the logistic equation that you can see here, where little n is the number of individuals here. Okay, and this k is known as the carrying capacity. Great, okay, that's easy enough. One little thing that I'm going to do is actually, for me today, will be convenient to define a relative abundance, which is this n, this number, divided by this long-term saturation of this species alone, so I'll call it capital N, the relative abundance. I will be sloppy with my language and use the word abundance for the relative abundance, okay? But that's all I mean. I will keep using the relative abundance throughout. And okay, the only difference is that now this capital N goes to 1 instead of to k. Okay? And the R here in the prefactor is the growth rate, okay? The initial exponential growth rate. Good. That was one species, but what our true interest is in many species. So for two species, what we will do is have the same logistic growth for each of them, but we will add an interaction. Now, this interaction that we're adding here in red, alpha 1, 2, N2, alpha 2, 1, N1, is an interaction that linearly changes the growth rate of the other species, okay? So if you look at the entire equation, it's a nonlinear interaction. You see here that N1 has R1, N1, alpha 1, 2, N2. It's a bilinear, N1 times N2 that enters the thing, like a mass action if you like, okay? So we have these two new terms here when we have two species. And now that means that we have two new parameters in our system, alpha 1, 2 and alpha 2, 1, where with the convention of signs that I used here, a positive number, it means that one species is deleterious to the growth of the other species. It's what happens in competition or predation. And negative numbers are when they're beneficial, such as in mutualism. Okay, so it's enough that we have these two numbers, and already by choosing the signs, we can have these different interactions that I described before that are written there on the right. Okay, so far it's clear. Yes, good. Okay, and what happens when you have many species? Well, you just do the same thing. Okay, so now we have S. Capital S is the number of species. Okay, we'll use that throughout. Think S for species, number of species. And for each species now, I have an equation for how fast it grows. And it's just as before, except I have one such term for each other species in my community. Okay? And the last thing I have is, remember that what I want to also add to my model is this idea that individuals come from the outside, okay, and in order to start growing in my local community. And so I'm adding this additional term here which describes migration, okay, just an inflow of birds flying onto my island.
Do you need that for stability?
So it's that's part of what I'll be talking about, what is and isn't needed and when and so on, okay? Right. Okay, so I just rewrote it now with a sum, okay, and this is it. This is the final form. And if you notice, when I changed it here, I had, oh, I forgot there should be a minus N1 as there was before, the logistic minus N1, minus Ni here. Okay, I forgot to add it here. So when you do add it, you have this matrix and you just call alpha II, the diagonal of this matrix, you just say it's 1. Now comes a crucial point in this whole discussion. And it just continues what we heard this morning, earlier this morning, that, okay, suppose I have, let's be modest, I have 50 species, okay? That means that this matrix has over 2,000 parameters, which I am not going to measure all of them in a field experiment, certainly not with a high accuracy. So I don't know the rules of the game. Okay, this is a weird situation. If you think of other fields, I don't know. If you think of mechanics, you know the rules of the game, and you're supposed to get some answer. That's your problem. Here are the rules of the game. Here's what I'm looking for. Here, you don't even know what the numbers are, what the parameters are. Okay, there's so much freedom in the interactions here. So I'm going to take this step of trying to use some simple random sampling of these numbers and see how far it takes me, okay? I'm not going to say, I don't want to say, okay, this is obvious, this is what you should do, you know. Let's do it and let's see what that does for us, okay? Why? Because there's nothing better that I should do, and certainly lots of the information in these 2,000 parameters shouldn't matter a lot. Then let's try to see how this goes along. Okay, so I'll call this the reference model, and it's the simplest model I can think of. I just take these different numbers in this 50 by 50 matrix. and just put independent random numbers, okay, all from the same distribution. And the only thing I do want to add is a level of symmetry, because I might want the effect of species I on species J to be correlated with the effect of species J on species I. For example, if I have competition and I want if this competes with this strongly, then this competes with this strongly as well. So this gamma, which is the correlation of the two sides of the diagonal, I might want it to be positive, or I might want it to be negative if I want predator-prey interactions, and so on. Okay, so yeah. And now the point is that my focus is on diverse communities with many species. So the limit I'll be taking, or where I'll be going, what I'll be thinking about is a case where I have many species. Okay, what is many? How much is many? That we'll have to see later. We'll have some results for very large S, and then we can test them on 7 species and ask, well, is it good for 7 species or not? But I'm leaving that for later. Okay, right. And as we'll see later, there's an important point here that we'll talk about, that there's a limit taken rescaling the variance of these alphas as I increase S. We'll talk about that. Okay, I'll keep emphasizing and stressing and saying again the same things. This is a very specific model. You know, I said, okay, let's for simplicity do this. Let's for simplicity do that. You know, maybe this. And then I took the alphas in one way or another and so on. This is definitely a specific model. And generally, there's no reason for this model to be an exact description of any ecosystem, of any ecological community out there. Actually, I'll show you later that there is one example where it happens to work very well, and another example I don't believe it works well. But the point is that hopefully some of the things that this model teaches us are more general, okay? And the question we need to keep asking is, what are the things that we believe are more robust, more general, more universal, and what are the things that are specific, okay? And if we don't ask that question, we're doing a disservice to this whole project. Okay, that's the crucial point here. What are the questions? What are the predictions that I can ask safely? All right. So why am I using these random matrices? People have suggested many such matrices of interactions. This is from something called competition colonization. It's a specific mechanism for species to coexist. And the idea is that if there's one thing that's very dominant in your ecosystem, there's one way by which the species coexist, and you understand it, then maybe a matrix like this works. But if you have many things happening, there's competition, and there's different, I don't know, again, go back to the ants. They're also cultivating a little garden, and there's some microbial population there next to the roots of the plants. Then who knows? Then let's take this. Maybe that's a better way to start. Okay, so now what I want to do is just, if for those of you who haven't played a bit with these systems, I want to give you just examples of what happens when you run the dynamics on a computer. Let's do a simulation. So what I'm showing here, remember we have S differential equations coupled together. So I put them in a computer. I run, this is x-axis is time, and y-axis are these n's, these relative abundances. As a function of time, each line here is a different color. And right, and so what you see is that some of the species have gone down very, very low and then got stuck here. At some low level, that's because of the migration. Basically, it's just individuals coming in from the outside. But they're not able to establish themselves as a big population. So they can't grow, okay? And up here, you see that there's another subpopulation which has established itself, and they have large relative abundances, much more than the migration. So they're living there, they're happy, and they don't need the migration in order to live there. There were questions, yes?
Quench dynamics?
Yes. So the alpha ij's once I fix them, they're done, okay? If I had, for example, evolution, I would start changing them. Or if my species would affect the environment in some way, which would then affect their interactions, then I might want to change them or add additional variables to bottle that. But all my alphas here, my R's, K's, all these things are fixed. Another question?
How do you select the growth rates?
The growth rates? Okay, so for most of what I'll be talking about, it won't really matter as long as, let's say that they're all positive. Okay, so let's assume, again, this also can be broken, but let's say for a predator, if it doesn't have the prey, it might be negative. But let's just take them all positive here. It would change. Okay, I'll try to remark where it changed things. Basically, if I show you all kinds of lines, it changes where these lines sit, but it doesn't change any of the qualitative behaviors that I'll show. Yeah, you can take them at random. You can. Sometimes you might have. This is the kind of data that you might have more of. Okay, because these are just S numbers as opposed to S squared numbers. So yeah, let's say you take them at random. That would be good enough for the discussion, in some limited scope. Anything else? Yeah.
You have to get the state states like that.
Yeah. Okay, so excellent question. I'll go on. Any? Yeah. Okay, so. That's not the only thing that can happen, but before that, so here you reach the fixed point of the dynamics. Each species is somewhere, and that's where it is, and it's not changing its abundance anywhere, anytime. Before I go on to anything else, even when you have fixed points, if you can restart the dynamics with different initial conditions, okay, with different initial abundances and see what happens. In some cases, you always reach the same final result of those are happy, those are unhappy, and the numbers are the same. In other cases, you reach different solutions, many solutions. It's possible to reach very many solutions in some cases. And finally, there might be cases where you don't reach any any solution for long times. And this is an example of a simulation with many species, but you can see 3 of them here plotted, and you can see that they're doing rather wild things here. And this is, in fact, chaos in very high dimension. Okay, so these are just options. And the fact that this is a difficult problem from a theoretical point of view, I don't want to go too much into it, but just think of this, even trying to find a fixed point of this equation, namely where species don't, where the abundances don't change anymore like we had here, in this case, then for this to be equal to 0, either this is 0 or this whole thing in blue is 0, or either the red or blue. And this is a hard optimization problem, right? You need to, or constraint satisfaction problem. You need to decide which subset of species is there. And once that's, and for the subset of species that is there, you solve this equation and all the ends have to be positive, okay? So you can guess that maybe species 2 and 5 are dead, but then species 2 was supposed to help species 4, so now that species 2 is not there, maybe species 4 is dead, but species 4 was actually, competing very strongly with the species 2, then it can come up and, it's a complicated problem. Okay.
So are there some statements about the uniqueness of things?
I'll get to that. Yeah, absolutely. I'll get to that. Okay, so we and other people have spent quite a while, squeezed this lemon and tried to get as much lemonade out of it, and really tried to study this model and understand it with as much detail. Not everything, but many of the important things. We looked at the properties of these fixed points. By the way, equilibria is in the sense of dynamical fixed systems. It's a fixed point. Okay, if I say equilibria, I mean at long times nothing changes. All the numbers are the same for long times. How many species can survive? What's the average of these abundances? fluctuations, things about the dynamics, many other things. And what I want now is actually to show you a sketch of a derivation that will show you how to get specifically what I'm showing here, which is the species abundance distribution. What is that? You take S species and you plot on the x-axis the value at n, and this is the histogram. Okay, so this approximates, of course, when S is large, this approximates the continuous probability distribution. Okay, so this is called the species abundance distribution. And by the way, the question of how big does S have to be, let's already look at it here. When I have 200 species, my species abundance distribution is this blue line here. You see it approximates very well the theory, which I'll show you in a second. But also for 15 species, out of which only 7 survived, something like that, if I run it many times, I get something that's very close. It's reasonably close. It's not perfect, but it's reasonably close. So you don't necessarily have to go to very large communities in order for this to be predictive. Yes?
But this is obtained in single equilibrium, so when you reach the fixed point.
Yes, yes.
In the single equilibrium.
Yes, yes. And what this is a prediction for the fixed point, and the simulations are at the fixed point. But I'll show you now really how to derive this from scratch. Any more questions up to here? No? Okay, good. So now let me try to do this. Here. Okay, so let me write down the equation of the dynamics, the equation of motion. So I remind you that I have these ni's. I is the number of species from 1 to s. And so I have that dni to dt is ri and I1 minus ni minus sum over j different from I, j different from I, of alpha ij and j, plus this number lambda here, which is the migration. It might depend on I, it might not, it doesn't matter. The important thing for me and what I'll show you now is that I'm interested in the case where these lambdas, this migration, is small. Okay, so lambda much smaller than 1, or if you like to be more formal and mathematical, lambda going to 0 plus. Small but finite number, okay? It's much smaller than all the other numbers that are here in this thing. And now what I want to write down is the way I'm sampling these alphas, these interactions. As I said, the Ri won't matter very much right now. The alpha ij's are, let me write it down in this way. Plus sigma over square root of s aij. Okay? So mu and sigma are fixed numbers, okay? Parameters of my model. And why did I write it with a 1 over s here and 1 over square root of s here? Let me say first, what is this aij? So aij are numbers from some distribution, and I take them such that the mean, so these angular brackets are mean, so the mean of aij is 0, the variance of the aij is 1, it's just normalized, and they're independent of one another. Okay, any two aij's are different from one another. Okay, and what is the point of this rescaling here by s and square root of s? What this does is I'm looking at a situation where I have colors, wonderful. So this is the term that has to do with the species interacting with itself, the self-regulation, if you like, or whatever you want to call it. This is the effect of the others. I'm interested in the regime where these two are comparable. Okay, I want to see things that are, it's not that it's only by itself, and it's not that it's completely overwhelmed by the others. And for that, if I have s terms here, and I add them up, then this scaling here would mean that the mean and the variance of this effect over the population will remain finite, will be of the same order as this. Okay, so you can see this more carefully, but I'm just trying to tell you the idea behind it. Okay, that's the point. Okay. By the way, you don't have to have this. This can be a finite mean, but this really has to have this rescaling here. Those are more technical issues. Okay, and I'm going to assume finally that all the Ri's are positive, as I said before. Okay, and now what I want to say, now I want to ask, I'm going to assume that a system or a community has reached a fixed point. Reached a fixed point. Okay? What does that mean? That means that these dynamics have reached 0 here. Okay? I'm at a long time. This has reached 0 here. Okay? And now my question is, for a large system, question, when S is large, what is the probability distribution of these ni's? Okay, I do the histogram as before. What do I get? Okay, and what I'm going to do now, the trick or the technique I'm going to use is something that goes by many names. One of them is the cavity method. And I want to give you absolutely the simplest version of it. And just to give you an idea how this thing works. So cavity method. This is a technique that was born, you can say, around disordered systems in physics, a bit of neural networks. Those are the areas where it was born 40 years ago or so. And we're applying it here. It was first, by the way, applied to models from ecology already in the early 90s, and it was completely ignored. And recently, people have come back to this. So here's the idea. So imagine that you have a community that has S species in it. Okay? You have N1, N2, N3, and so on and so forth. And now what you're doing is you're saying, let's imagine that a new species has come into the community. Okay? So that new species, let's give it a different name. Let's call it, its abundance N0. So its index is 0. Okay, so now I have a new community, a larger one, that in total has S plus 1 species in it. Okay? And of course, this N0 here interacts with all these different other species here. Okay? So I have for each one of these new interactions between N0 and N1, N0 and N2, N0 and N3, I have alpha 0, 1 and alpha 1, 0, which is the effect of 0 on 1 and the effect of 1 on 0 and so on. And the crucial thing is that these new alphas that I've added here, alpha 0, 2, alpha 2, 0, and so on and so forth, these are new numbers that are completely uncorrelated, independent of everything that happens in here. These n's, I don't know, maybe they're strongly correlated. I don't know what's going on in there. But these numbers are definitely new numbers, you know, straight from the bakery of random numbers. Okay, so at the fixed point, I added this new N0. So I know that dN0 dt, I'm assuming that it's still at a fixed point after I added N0, is equal to N0, 1 minus N0 minus sum over I, 1 to S, alpha 0 I, N I. Okay, so this is the equation for this new species plus this small migration term. Okay? So now because this lambda, this migration term is going to, oh, sorry, yes?
Yeah, I'm sorry, I don't understand why it would be zero. Like, if we're adding a new species, why is it still at a fixed point? Wouldn't it change, or am I kind of conceptualizing it wrong?
The fixed point would change.
Yeah.
Yes, the fixed point would definitely change. So there's but what specifically is the question?
Okay, so what you're saying is we add a new species and then it changes to a new fixed point.
Yes, the whole thing changed. Okay, so let's break this into a few. Let me make a few points here, maybe, because this is a very observant question. So, indeed, two things. I haven't talked about this at all, but right now, but indeed, two things happen. Both this n0 is affected by these other species, but the whole fixed point might change. Okay, the idea is that because I have many numbers here that are quite small. The effect of this n0 on these n's is small. Now, that does not mean that, that you don't have to take that into account. This affects them, they're changed by a little, and then they affect this back, and so on and so forth. Closing this back and forth, it turns out that the change in these n's is not, you don't need to take that into account when these numbers Iij, Aij are independent. Okay? When you do have correlations between Aij and Aj, you do need to do another step there. Okay? I haven't even gotten to saying all these things, but it's good to say them right now. Okay? That's an expert question. Right. Anything? Sorry. Let me see. Okay. Good. Yeah. There was another question there.
Yeah. Still regarding that, you put the identity of n0 equals 0, right?
Mm-hmm.
Why?
I'm assuming, this is an assumption, that both before and after adding this new species, I'm at a fixed point.
For the other species, but why?
Also for the, so this one would, I'm assuming, it's an assumption, I'm assuming it would grow and be added to this fixed point. Okay? By the way, it might grow. So this is n0 as a function of time, and this is time. It might grow and reach some number, or it might crash down to 0, okay? But I'm assuming it does reach some number. Okay, anyone else? Good. Okay, so this is my equation. Now, remember, I'm going to take lambda very small, so let's look what happens at lambda equal to 0. For lambda equal to 0 here, either n0 is 0, which means that the species has gone extinct. This is this case here. Or n0 has to satisfy this equation here. Okay? Okay, so let me write it this way. So either n0 is equal to 0, for lambda equal to 0. N0 is equal to 0, or n0 is equal to g. G comes from solving this, where g is just 1 minus this thing here, okay? So all this equal to 0 means that g is equal to 1 minus this sum I1 to s alpha 0i and I, okay? So those are the two solutions, okay? So now, what do I want to say in this? So if g is negative, this number can be either positive or negative, right? If g is negative, then n0 cannot be negative, okay? These are abundances. They're number of individuals, so it can't be negative, okay? So that rules out that solution, and you're only left with a solution n0 equal to 0. So that's one option, okay? If g is smaller than 0, then n0 cannot be negative, and therefore n0 is equal to 0, okay? Actually, because there's this little lambda here, n0 is a bit above, okay? But it's still small. Another option, or another situation, is where g is larger than 0, then it looks like there's two solutions. Because n0 equal to g is now okay, because it's a positive number, but also the 0 looks like it's okay. But it turns out that this solution then is not stable. Namely, if it starts very low, it would grow up to the other solution. Okay, so in this case, the solution is n0 equal to g. Okay? So let me put it all together. Putting these two together, I get that n0 is equal to maximum of 0 and g. Okay? Good. Yes?
Since you have the first constraints on the abundances, why you don't get a delta peak in the distribution in the theorem? Because you
I will. I have a bunch of numbers that are going to be on 0. I'll get that. Okay. Yeah, sorry, this is an excellent thing. What I showed here in this distribution is only the species that are alive, surviving, coexisting. There's still a bunch of species that are just dead, and then I should get a peak that I did not draw here. This is a very good, thank you for this remark, okay? It's clear what I'm saying? So these are the 40% or however many that did survive, and there's a bunch of them that are dead, and I just didn't draw them here. They're all on 0. Okay, thanks. Okay, so now the next step is I just rewrite G. So G is this, I remind you that this is G here. So I'm just rewriting G. I'm plugging in this thing for alpha, okay? And I get that G is 1 minus mu over S sum over I equal to 1 to S of Ni. And this is equal roughly, there's N terms here, there's S terms here, so I replace them with the average times S. And divided by S, I'm just getting mu times the average of Ni. Average of Ni is any Ni here. All of them have the same average, okay? It's just the average of all species. That's just a notation, okay? And so this is one term minus sigma over square root of S sum over I 1 to S A0 I N I. Okay, and that's the second term. Okay, so now what I can do, I'm not going to do it now, but it's a straightforward exercise in taking the mean and the variance. I can take the mean and the variance of G. Okay, I take the mean, and the mean here, for example, I'm just giving you an example. Take the mean of this thing. This isn't just a number, and here the mean of this thing here, A0I, will be 0. So I'll just get 1 minus mu Ni. Okay, so G average is 1 minus mu mean of Ni, and I can also do the variance of G, and I get, if you do it, sigma squared Ni squared average. Okay, do the calculation. Okay, and now comes the final important point of this whole exercise, and it is the following. So this was my relation between N0 and G, okay? G is a sum of many, is 1 minus the sum of many numbers. These alpha 0I are independent. This Ni makes them slightly less independent. Bottom line, you can show that this thing is a Gaussian. Okay, this is a Gaussian distribution, this G here. Okay, this Gaussian distribution has a mean and has a variance. These mean and variance are functions of what? Their functions are our parameters mu and sigma, which is function of the interactions that we assume, and they're function of the properties of these n's. So as a function of the properties, the mean and variance of the existing community, and the properties of the interactions, I get a Gaussian distribution for this G, and N0 is then just given as the maximum between 0 and G. Okay? So what I know already is that, as a result, N0, P of N0, will be a truncated Gaussian. It will be G is this distribution. Out of it, the maximum will only pick the cases when G is positive. Okay? And as Ada remarked correctly, there's all the species that are dead, and they're all sitting here at 0. Okay? So the last thing I basically need to know, I know almost everything, I need to know just one last thing, And that is, I need to know what is the width and the mean of this distribution, right? And if I know that, I'm done, okay? So now comes the last crucial step, and that is saying the following. Okay, I added this, I had this procedure, I had S species, I added this new one, and now I have a larger system, okay? But now this larger system, I could think of it as a system with S plus 1 species. Nothing is very special about this N0 that I added once I added it, okay? So it has the same properties as these species here, okay? It's just one of them, okay? And therefore, the last thing that I need to do is say that N0 average, I require that it be equal to the average of the other ni's, and N0 square, I require that it be the same as the square of the others, okay? Those are requirements, constraints. I have to have that. And then I'm done, because then what I can write that this N0, for example, this thing, is equal to the integral of P of G, G dg. Okay, it's basically, it's when do I get, what contributes to this mean? All these zeros don't contribute, only the positive part here contributes, so it's this thing here, okay? This P of G, I know it as a function of these ni's, okay? So here, this thing depends on mu, on sigma, on ni, and on ni squared, okay? So this is an annoying error function that depends on all the parameters, but it has to be equal to this thing here. So I have a self-consistent equation for these parameters, for the ni, and another one is the same thing. This thing here is equal to the integral P of G, G squared dg. Right, and these are two equations for the unknowns ni and ni squared. Okay? I can solve them in nonlinear. It's possible to solve them. It's not a hard procedure. Believe me that this is easy to solve. Okay? So by that, I got also the width of the distribution, and I know everything about it. Okay? Yes, questions?
But if you have no fixed point.
Right, okay, we'll talk about that. No. I'm just trying to give you the flavor. There's so much here that I didn't tell you. And I'm trying not to cheat, so I'll just say a few small comments at the end. So you know, legally I am covered. One of them, as was mentioned before, the ends here are also. I already said this, but let me say this again. If these ends are, they're shifted a bit by the introduction of this one, and you might have to take that into account, and then it's a slightly different. If I had 30 more minutes, I would show you even this version. That's relevant when the correlations are, when there's correlations along the diagonal, more symmetry. Another thing is that, okay, just a comment. Note that the fraction of surviving species is just the integral of this curve up to here. This is integrated, this is 1, so it's just this part, and this part here, those are the species that did not survive locally in the community. And another extension is, an important extension, is that I could have done all this story in a time-dependent thing. So looking at the entire history as a function of time, adding another species that it too has a history as a function of time, and doing these effects back and forth. This is known, goes by the name of dynamical mean field theory. Okay? And it is useful for what I'll show you now. But I'm not doing it here, I'm just giving you the idea. Yes?
Why did we introduce the migration rate if now we consider it to be 0?
Where did the migration rate enter? Question to the crowd. Where did the migration, it entered in just one place. Any ideas? There was one place where I used it and then I took it to 0 and I didn't tell you. It entered here. When I said that here there's two solutions, but I choose one of them, the little migration that was there forced me to say that if a species has a positive growth rate, it's not going to stay low, it's going to go up. It doesn't matter how little that migration is. I could have, one parrot coming from India to Israel once every 10 years, And that's enough for them to fill the Yalcon River Park now, because we're getting close to, at least in terms of temperature, to conditions from other parts of the world. So that's the only place it entered, okay? But that's an excellent question. Anyone else? Yes?
Now the relative abundance, they are relative to what? Because you define it as, you know,
No, I started with this equation, which is already relative abundance. It's without the Ni. Yeah, so I didn't even look at the carrying capacities here. It's already, the equations are already divided. The variables are already divided by the.
But what's the meaning of the carrying capacity in that case?
No, it doesn't. It's out of the question. It was out of the equations already. I'm working with, imagine that I'll show you an experiment where we actually do this. You measure, I don't know if I'll have time to show you, but it's okay. But we actually measure the relative abundance. We measure both the carrying capacities alone and in pairs or more, and we just do the division, and we assume that after that, this is the equation. So yes, the carrying capacities are out of the game.
But how do you measure the carrying capacity?
You can measure them, for example, by growing things alone, and then growing things in pairs or in 4 species, in 8 species, and so on.
Suppose that the things can grow alone.
Yes. So that was another assumption. I'm covered. I'm legally covered here. That the Ri's are larger than 0. That might or might not be true. If you take microbes from the ocean, many of them you cannot just culture them. Certainly not in the conditions that you have in your lab often. For plants, it's a reasonable thing to do. Okay? And if you do experiments with culturable bacteria from the beginning, then you are in a good place. So it might or might not be a good place to start. I agree with you. Yes. Mm-hmm.
Why can you have abundances that are higher than the carrying capacity?
Why can't I?
No, why does it occur?
Yeah, because some of these numbers might be negative in. I didn't say how I take the distribution of these numbers. Okay, if they're negative, then yeah, so I have no issue with that. But that's a good point. Yes? Anything else? Okay.
I was just wondering how, because all of this analysis is based on the fixed point. I mean, you start with the fixed point equation. And I was wondering, what does this tell you about actually the number of steady states that are?
Okay, wonderful. You're starting to encroach my talk, so maybe it's time to move on. These are excellent questions. If by the end, which I won't get to anywhere near it, you have them, please let's have that discussion then. But this is really the mere nothing, so you understand what we're talking about. Okay, good. So let's go on. Excellent. So point is that one thing that I didn't even say here, but now you can notice it, that I never told you what the distribution of the alpha ij's is. It could have been a flat uniform distribution, Gaussian, who knows what it is, okay, something else. As long as it's not very crazy, this never entered here because of something like a central limit theorem. So the important thing is that the distribution didn't enter, and that means that really what I have is 3 relevant parameters in my system, okay? And these are this mu here that I showed you before, this sigma, which is not exactly the sigma here. This is for a slightly extended case. It doesn't matter right now. And this correlation that we discussed before between this level of symmetry in the system. Okay, so a few remarks. So this was, again, a very specific case. And one of our themes is that we want to understand how robust things are. We're not trusting Lotka-Volterra. We're not trusting our ecology on Lotka-Volterra, okay? So one thing that my collaborators in Michel Laurent's group, Mathieu Barbier and Jean-François Arnoldi have done is taken many different options for how to sample these alpha ij's from the literature, common things, small world networks, resource competition, all kinds of things if you know them, and I've tested all kinds of effects, and most different options for sampling things don't change things very much. What can change things is, for example, if you have two groups, two functional groups. If you have bees and flowers, then saying, okay, I don't care, the bees compete between them, the flowers compete between them, and the bees and the flowers help each other. So saying, okay, all of them are a bee flower and I just mix things up completely, that doesn't work. In cases like that, where you have functional groups, you can extend the model to a model that has groups, and each connection within the group and between groups has these parameters, has something like these parameters. So it's an extension that then you can do all this kind of theory, and then it works very nicely. Yes?
Is this result somehow an extension of May's theorem?
May's theorem takes us to a very different place in life. Let's do the unlearning now, and we can talk later about May, okay? I really don't, that's a bad place to start. Okay? No, it's a wonderful thing, but it's just, anyway, let's not discuss that now. Okay? It's not nothing against what May said. It's against trying to throw everything on that one thing. That's what I don't like at all. Okay, good. So going back to this position in ecology, we said people are doing complex simulation, getting a lot of insight from these few species models. What we're suggesting to add here to ecologists is these simple large-scale models, which include all these species, include this heterogeneity in interactions within functional groups, but are simple enough, and so you're not throwing the baby with the water. Okay, so that's a message more to ecologists, if there are any in the crowd. Okay, good. So let's go on. Good, but now I want to do something else. Now I want to say, look, now we have three numbers. Remember, mu, sigma, gamma, which are all I care about. We can start plotting whatever we want in these three numbers. These are my temperature, pressure, and density if I think of a gas. Okay, that's what I want to think about. And then I can start plotting phase diagrams, as we call them in physics. And what I see here, I'm plotting, let's say, the mean alpha against this number, which comes from here, for the sigma here, essentially. And what I see is essentially, I see a few phases. And by phases here, I don't mean like those who studied equilibrium statistical mechanics. I don't mean water and gas. I mean qualitatively different behaviors of the dynamics with sharp transitions when I change the parameters. Okay, that is what I'm talking about. So there is a regime of parameters. Here, this triangle. This triangle is exact, by the way. It's a straight triangle, where I get this thing about the fixed points, which I discussed so far. What I did down here is basically valid here. Then I also get a regime where I had this, I don't have it here. Yeah, a regime where I had up here, I get this chaos. If you remember, these abundance is going up and down like crazy, doing all these crazy fluctuations. And then I get other stuff that I'm not going to talk about. So what I want to do now, yeah, I saw you, just a second. What I want to do now is, because I don't have time and I don't want to throw on you too many things, I want to just show one transition, one change from this behavior, this fixed point behavior to the chaos, and really explain what happens there, and then show you some comparisons with actual data. Yes, there was a question?
Yeah, sorry to interrupt you again, but I'm not sure. The results here are just from simulations or also from theoretical?
This is also from theoretical stuff. It's analytical. It's fully analytical. So this gives you certain things. The extension of this to dynamics gives you things about the dynamics as well, and when fixed points are unstable, and so on and so forth. Okay? Okay, so let's dive into just one transition, one change from the unique fixed point. So what happens? I start with species, and at the bottom of this line, this y-axis here is either the variability in the interactions, how much certain interactions pairs are stronger and others are weaker, or growth of the number of species. So I can do both. I can have a community in an experiment that has more and more species, or a community that has more and more variation in the interactions, some strong, some weak, and so on. When they're all more or less the same or have some variation, finite variation, I get this fixed point. In this fixed point phase, okay, when there's no variation, all of them survive and coexist together. When there's some variation, some start going extinct. I know this. I've calculated it before, okay? I know why some start going extinct. This is the first thing that happens. It turns out that I'm still in a fixed point. Here, you see the simulation. We understand this analytically. I'm still in a fixed point. So this is this point here. Then at some point, I reach this point here where my fixed point is no longer stable and I start seeing fluctuations, okay? As I keep going up along this line, these fluctuations grow, okay? Just the size of the fluctuations, if close to this transition point, I see that n as a function of time makes small fluctuations. As I go up, it makes larger and larger fluctuations. Okay, so that is the scenario that I see in this model, and actually in models including resource competition and a bunch of other things. So this, we hope, is more general than the specifics of this Lotka-Volterra model. Okay. Now, I want to say that although some things that are happening here are not obvious. It didn't have to be the case that first species, as I start increasing this heterogeneity, that first species would go extinct and then I would lose stability and start seeing fluctuations. It could have been the other way around. It could have been that species would go extinct so fast that I reach a few species and I never see fluctuations. Okay, for example, all these things are things that are actual predictions that I can hope that will extend beyond my very simple model that I used on here and down here. Anyone here who has May in their mind, May has to do with linearization of these equations. Okay, all these things here about extinctions and fluctuations, these are fully nonlinear effects. Okay, so all the predictions I just mentioned now are fully nonlinear effects, yeah, that's where they come from. Okay, good. So now let me jump to show you some very recent experimental data. Okay, so what is it we want to show in an experiment? So just a second. I talked to people, Jeff Gore's lab at MIT, and he said, okay, good. Supposing I do the effort and put students on this, what is it that we want to show, right? All this is very nice, but what is it that you want to show? Okay, so what are the things that are new here and possibly interesting? So people have recorded fluctuations in natural environments, that abundances, that some species go up and down and so on. But as I mentioned, it's hard to say what the source of these fluctuations is. Sometimes you can argue that it is internal, inherent to the system rather than external fluctuations. But even then, how generic is it? How much do I have to fine-tune the system? Is it something very specific, or will I see it in many places in nature? So one thing is I want to have a unified picture where I see in one experiment both the fixed points and these fluctuations and all these different scenarios in a controlled way. Another thing is, if I see something like the Lotka-Volterra in an experiment, that is going very much towards this idea that this is not specific to the types of models that I looked at, that this has relevant to nature, okay? So those are the two things that I have to say, but the thing, again, I keep saying again the same thing, the thing that I that I believe is extendable is not the details of this Gaussian and so on, but this scenario that I just showed you before about how the order of things and how they happen. These are the things I expect to. This is not easy. These are not easy experiments and are not easy to see. As I said, natural systems, you always suspect it's external conditions. In experimental conditions, you need to control the strength of interactions, the variability of interactions somehow. Okay, so this is there's something here to do. Okay, so now let me go and show you. This is really recent stuff. I'm supporting theoretically, but this is really work from Jeff Gore's lab at MIT, as I said, with Jiliang Hu and Daniel Amour and Mathieu Barbier, who's actually here in France. And the idea is the following. What they did is they isolated species from worm gut microbiome and from soil. And what they did is a simple protocol where they put a subset of the species into some growth medium, and they basically just do daily dilutions for 10 days and just see who's there. They can sequence and see who's there in it one abundance with time. They measure, sequencing, biomass, pH, and that's about it. Another thing is that I won't get into the details of how this works, but what they can do is through a mechanism which I won't talk about, they can control the strength of interactions. So forgetting about the biology, the nice thing is that we can measure the strength of interactions with doing pair species experiments. You put two species in this medium, you measure and you see, well, did one win, did the other win, did both coexist? We have information which is buffered, pun intended, from the biology. Okay, so we really, we could really say, okay, if this is a Lotka-Volterra model or something like it, the strength of interactions, the variability of interactions has increased as I changed conditions. Okay, so we have the two knobs that we actually want the most. We have the size of the species pool. They can add anything from, they have now up to 50, now up to 100 species, and they can change the interaction strength or interaction variability. These are simulations, again, from Lotka-Volterra. I'm showing you, it's the same thing as the phase diagram I showed you before. I'm just doing it in the experimental knobs. Okay? So these are the following things. Let's start from 3. 3 is the regime of parameters where you expect to see fluctuations around a fixed point. Both regimes 1 and 2 are fixed point places. But remember that we said that as we go into this regime, we're already seeing extinctions. So this dashed line is where you see the first extinction. Okay? So there's 3 different regimes. All species survive. Some species survive and reach an equilibrium and fluctuations. Okay? Okay, and then they run combinations of 3, 6, 12, 24, 48 species, subsets of species. And for each experiment, which runs for 10 days, 11 days, they ask how many species survive at the end, and do abundances fluctuate. So this is the total biomass, and you see, and this is the number of species in the original pool of species, and this is this interaction strength, which they can control. And I want to say that there is, just like here, a small migration. Okay, every day they add just a little bit of everyone to give everyone a chance to keep growing up again.
So even the ones that went extinct?
Even the ones that have gone extinct are given the chance. Okay, they might not be able to use it because they have negative growth rates, but they're given the chance to try and come back up again.
I think I have a question precisely about that, because intuitively I would think if we wait very long, then and we're in the very high fluctuation regime, everyone should go extinct.
Not everyone, but without migration. So what I was discussing the whole time.
Everyone but one who then has nobody to interact with, and then the algorithm.
So without migration, that is correct. Well, yes, to 98% you're correct. But everything I said here is with small but finite migration, and then what happens is that species crash down. You had this toad, and for 20 years you did not see it, and then you see in the news, this toad was rediscovered after 20 years. That's because it was there, it has negative growth rates, and then all of a sudden it picked up for some reason, which could be the other species that have changed in that park. Okay, so yeah, so what you see is that the fluctuations are measured in origin, those that are fluctuating at the very last 4 days, and you see that for low interaction strength, many or all survive, and they're not fluctuating, whereas as you go up in this direction or up in that direction, you see more and more fluctuations. Okay? And basically taking this data and data about the, just this which species survived, which is sequencing data, simple sequencing data, this is the experimental result here. Again, number of species and interaction strength. And this is the fraction of species that survived, namely that are in significant abundances at the end of the experiment, between 1 and 0. And you can see that there's This dashed line is where all species survived, and then you see less and less species surviving up to 0.2, roughly a fifth of the species survive in the large thing, in the large S's and large interaction strengths. And the second thing is the fraction of the communities which were fluctuating between days 6 and 10. Okay, so you waited a few days because always at the beginning there's things rearranging, but then those are those that fluctuated. And so the solid line is this line here. So you see the comparison with the simulations, okay? What is it that we believe and what is it that we don't believe? Let's start with what we don't believe. We don't believe the numbers. The numbers here do not correspond to the numbers here. The numbers here don't correspond to the numbers here. What is it that we are gaining? We're seeing the order of the behaviors transition in the order that we expected. And we're seeing that as you go up to higher interaction strength or across increasing the species pool, you're getting the same series of things happening. So yeah, so this is an experimental phase diagram that we are getting. Okay? Questions about that? Yeah.
All the model deals with abundances that are just normalized by the carrying capacity.
Indeed.
But actually, when you do the 16S sequencing, you don't have this kind of abundance. What you actually measure is the relative abundance normalized for all the species. Different kind of normalization. I don't know how much it affects.
No, I agree 100%. That's why I did not try and am not pushing for and am not arguing for getting to get the species abundance distribution. That is not something I would show you now, okay? And a bunch of other things which I wouldn't show you, okay? So again, the question is what questions we can and what questions we can't ask and hope for them to be robust, okay? Yes?
Just a quick question, experimental question.
Mm-hmm.
When you do these measurements, how do you know that the environment is constant? How do you know that you know that you've reached a steady state that is not due to, for instance, the deprivation of the nutrients?
Okay, so first of all, these are cycles of 24 hours, okay? And you might or might not reach stationary phase of the bacteria, for example, in that time. I am not even, we happen to know if you don't or we do. I'm not even committing to that. That is not, that is not the issue I'm trying to solve. I believe that we haven't done this, but I believe that if you would take, faster, more frequent dilutions cycles, and you would be in an exponentially growing phase all the time, it would still be very similar things. The point is, to me, more that you waited enough time so that as a community, whatever the dynamics are, for between days 6 and 10, that you're seeing more or less the same thing. And if you look at different statistics, you are stabilized at the level of the community, whether it's fluctuating or not, okay? So yeah, so these are very specific conditions. It's not, the point is not to try every type of condition bacteria might live in.
If you want to do the matching between the theory and experiments, you need to make sure that experimentally you are actually looking at things.
Let me tell you even worse. I'll open the door to tell you it's much worse than what you're saying. These are continuous time simulations. This is a dilution experiment, okay? We did try, by the way, to do simulations which are dilution simulations. If you look at within the 24 hours, it looks so different. But if you then look at the order of these phases and when you grow in this direction and grow in that direction, how the lines go, these things don't change, okay? That's my message again and again, I think. I realize that I'm just giving you one message, and that's fine. Anything else? Yes?
Can you make predictions about the extensivity of the fluctuations? Because within the fluctuating phase, you can have many different degrees of chaos.
Yes. The one thing...
You're considering it as a single phase. But
so the one thing that I can definitely say is that it would grow, and it would grow continuously. Okay, those are the two things I want to insist on. Okay, it grows and it grows continuously, and by continuously I mean once you cross the transition, it's not that you jump in the size of the fluctuations. But to the best of my knowledge right now and understanding of what happens theoretically in this phase, there is no additional qualitative jump. There's not an additional phase in there. There is a change in the strength of fluctuations, as I said. It's a big change, but it's not, there's nothing new qualitatively there. Okay? Yes?
What about the alpha ij, alpha ji correlation instead?
Yeah, so if you, this thing, and when you measure, it doesn't look like the matrix is symmetric. So we're not even, it's not relevant to this experiment. There's interesting theory, and we can discuss theory, if you like, over lunch, of what happens when it is symmetric and isn't symmetric, and there's lots of refinement there. Okay, I see I have 10 minutes, so I'm not going to tell you at all about coexistence and randomness, and I'm very sorry for you. But let me just close by telling you, just in 2 minutes, and then maybe if you have more questions, about different extensions, different settings that are very interesting, both theoretically and for these experiments in natural communities. So one thing a lot of people talk about is resource competition, where you actually say, okay, where are these alphas come from? So definitely in microbial systems, often what's happening is that microbes secrete something and then another microbe takes it in, which might be cross-feeding, it might be antibiotics, it might be a bunch of other things, okay? So you can write models which are explicitly putting the chemicals in. And what I want to say about these models in the present context is that they have the same transition to chaos and all the phenomenology I was talking about so far. So it sounds like it's a very, very different model with different assumptions. Bottom line, for us in this level, it is very similar. Another thing which is very not similar is adding space to the system. What do I mean by space? So far we talked about one location in space, and we said, well, there's some general place which I'm not specifying that's, bringing in, supplying the species. When you have explicit space, it turns out that this is a huge perturbation to our thinking, okay? And for example, in that case, you can have a chaos which is long-lasting and saved by the different places. So there's no longer this deus ex machina place which saves the species. Only a subset survives, but the subset that survives can survive for very long times that way. Okay, this is known as the rescue effect, and it turns out that that allows, for example, to go to diversities well above what you would get in fixed points. Yeah, so that's one thing. There's another very different thing that happens with space, and this is now my favorite pet right now. There's a case in different set of parameters. It's sparse interaction matrices. I'm not getting into it. The point that happens there is that when you attach places in space, each place reaches a fixed point, but because of migration, they can infect each other. They can convince the next place to get their subsets of species, okay? And then you can start having domain growth things, like where you have a spread of your subset of species going elsewhere, okay? And it turns out that there's even a function, which is the function that says which subsets of species, which solutions will be the ones that spreads through space, and then it acts like a fitness. So there's a level, at the level of a community, there's a Darwinian selection that we see, and we see how it promotes the growth of this fitness function. So it's really thinking of the entire community as an organism, but in a very systematic way. Sparse interactions, again, unlike resource competition, sparse interactions are a whole crazy thing. It's very different from what you've seen here. Yeah, that's one thing that's very interesting. And the last thing is noise, which I haven't talked about at all. Okay, noise is of course very important, both from the environment, which changes the conditions, changes the growth rates, and demographic noise when you have small numbers, all these things. you know, are interesting things that, you know, I'm happy to discuss. Yeah, okay, so I'll leave you with this. Yeah, I'm not going anywhere else. If you have any more questions. Yeah. Questions?
I can start with your first one for now. So you said that one of the things that you wanted to test with the experiments was whether you're at all in the Lotka-Volterra regime.
Whether, no, actually, whether the phases in their organization are the same. So the details of Lotka-Volterra in this experiment will probably not be
Okay, but I
validated.
think my question is broader and sort of back to your first point that you know, resources don't matter, but so there's also models with nonlinearities in the interaction terms. Does that matter, or basically, you know, for the questions of stability and all this that you're looking at, is everything the same as Lotka-Volterra?
No, I mean, no. So just like if you have, I think of it as really as we think of phases of matter. What's a phase of matter? It's robust, but not infinitely robust, okay? So I can have water going into, I don't know, a liquid going into steam, water going into steam, and I can have dirty water, and can have clean water, and it would do that, okay? But not everything. If I take something else, it would not do that, okay? So there's a finite thing, and the thing is to classify the other behaviors. So what I was trying to say, for example, with the space and the other things, is that there are limitations to these things, okay? But yes, a level of nonlinearity would not destroy this. Some finite level of nonlinearity of different kinds would not destroy this. It is robust at that level, okay? That's as much as I can argue for.
I'm sorry, I'm curious about the experiments. So like this interaction strength, fraction of surviving species, like fluctuations. Like, I mean, how did you measure all this?
So wait, let's go back to the experiments.
A little lost. Yeah.
This part? Yeah, so again, so you sequence, you take a sample from every day and you sequence it, okay? And that's how you know relative abundances, not absolute abundances. You have optical density, so you know the total biomass. Well, it's at least some measure of the total biomass, okay? Again, remember what we want. We just are asking more if it's fluctuating or not. Whether we're doing the absolute, or relative is if the optical density is exactly the biomass or not, is not so crucial to us, okay? Anything else about that?
Yeah, like the interaction strength.
Yes, so,
3 levels. So
right, so interaction strength, what you do is you do, no, so we didn't do it for all, so I didn't do anything. They didn't do it for all. So they took a subset of 6 species and did all 6 times 5 pairs. And what they saw is, what they looked for is whether both species coexist, one wins over the other, or whether one wins or the other can win, what's known as bistability, okay? And those things are enough to give you a measure of, so here what's plotted is the fraction of alpha larger than 1, which is what happens when at least one of the species dies. Okay, so you see you're changing the conditions, and you're seeing that the fraction of pair experiments, of the 30 pair experiments where one of the species dies, that grows. And the fact that it has to do with nutrient concentration, that's a complicated thing that, I'm happy to tell you, private if you like. But again, we don't need to understand, we don't need the biology, it doesn't enter our thing. We just need the interaction strength, some measure of that.
Any other question?
Yeah.
Is there any hope to get, let's say, to count the number of equilibria that are there in this experiment?
So this is very not symmetric, and there's 0 equilibria. This thing is fluctuating like mad. So yeah, that's the short answer. But in other cases, I believe that if there would be, you could see these things, definitely. You restart the system again and again, and you see if there are multiple alternative stable states. That's a big question in ecology, with or without us. Yeah.
All right, maybe it's lunchtime.